All right. So I think um, this is a last video lecture that we're doing for this course and for uh, doing some expression analysis. So reminder that we have files that are thousands of lines long that we really can't look at individual lines one at a time to really get an idea of what's happening in this data. So it almost becomes a requirement to plot some of this data out. Um, now, so this DESeq package already comes with a handful of plotting tools. Um, so we've already done this. We've already done the plot MA, right? So we've done this. We're looking at the normalized counts against the um, log fold change. So it kind of just tells us which genes or at least tells us how, it visually tells us how the percentage of genes that are differentially expressed, at least in a statistical framework. Um, there are a handful of other kinds of um, plots that are already pre-made into the DESeq package. So we can directly compare expression values um, for a single gene. It lets you do that. It already kind of works a little bit with GGplot behind the scenes. Um, so that also means that we can replicate a lot of these graphs by ourselves, assuming we know GGplot well enough. But just a couple leftover plots that can kind of help us out with understanding our data that's not are these heat maps. Um, uh, this more or less comparing samples to each other. It's more or less a visual way of measuring how similar two samples are to each other, and then comparing um, plotting those metrics of similarity out in a pairwise fashion. So this is one thing that we'll try today. And another one is a principal component analysis. So a principal component analysis is it's a multivariate statistical test. Um, you usually learn more about it in a graduate level stats class, but a principal component, what it does, so we have again 20,000 different columns, 20,000 rows, 6 columns. And so what a principal component analysis does is that it tries to summarize as much of the variation we can observe in our data into as few axes as possible. I know that's very weird to explain, but it also has what we can use it for is more or less clustering. So what you would expect in principal component space, similar similar samples will cluster together in principal component space. So this is one thing we'll practice with today. This again, today I'm just going to keep talking until I'm done with all of this, however long that may take. I don't think it'll take longer than an hour, but honestly, who knows? So I've walked through this already, and so just in my R environment, I loaded up everything um, to the results and wrote out the results. So I basically reran my DESeq analysis to regenerate these variable formats and these structure formats. So the first thing, right? So plot MA already exists. We've already done this plot MA, and so this we'll work on a PCA plot given our data. So again, a PCA is much easier to do on a computer than it is to explain what's happening behind the scenes. Like so many of these tools, it's very much like the analogy of a car. Like I kind of know how a car works. I definitely can't put one together, but I can drive it pretty well. And that's kind of what we're doing here. I kind of know how it works. Couldn't build it myself, but I know how to use these tools. Um, So whenever we plot this stuff out, one of the first things that we have to do is transform our data. So these raw values, they range from like 0.001 all the way up to 100,000 plus. So our data is already kind of on this um, log data scale. So we need to transform our data into this VST is basically doing a transformation. It's doing a log transformation. So this VST is a built-in command to uh, uh, the DESeq package. I think I set that to false. And so what this is going to do is just going to make a variable, and it's going to log to transform all of our values. We can do this ourselves, but this is kind of a wonky data structure, and the plot PCA plot is a little wonky itself. We can extract these values and do our own principal analysis and plot stuff out in GGplot. But for now, that's kind of tedious. We don't want to do it when we can just immediately look at it. So let me just call this just just a variable. It's just a transformed data set. Um, I don't know how long that didn't take long at all. So again, the plot PCA is really easy. It's just one command. It's calling this transform data set um, in group. So this is like labeling um, colors of our samples based off of species and the tissue. So this is very easy. And what this plot shows, and it definitely is the expectation. So if we're looking in principal component space, um, all of the kidney tissues are more similar to each other than anything else, and all of the liver tissues are all similar to each other, and they're more similar to each other than anything else. And there's really not a whole lot of overlap between these two tissues, given overall expression patterns. Um, Again, these plots are going to look really, really nice because we're working with different tissues. But if we're looking at an experiment where we have treatment groups versus a control, these everything may not look always so clear all of the time. It's just how our data looks. And so the other one that we'll work on based off of what's available in the DESeq package is by comparing, kind of measuring the distance of these data sets, kind of in the similar way that we can estimate a distance between two nucleotide sequences. We just compare expression values for every single gene and measure distances, pairwise distances for all of those gene numbers, and then kind of come up with an overall mean distance um, of those values. So this one takes a little bit longer, and it's not because it's hard, it's just kind of tedious to get this plot the way we want it looking. But again, all it's doing is just comparing distances. And so the only command that's doing this is this sample dist command. So again, nothing in R is intuitive. And so I don't know how this command is working, but what I know it's doing is that it's just measuring the distances among the gene expression values of these log transform data. So I'm going to write this. I don't know if I necessarily want to write this out. It is a decent amount of code. Um, but yeah, that's measuring the distance. This line is just converting it into the proper kind of data frame as a matrix. So we're making sure that this is saved as a matrix and not as a data frame or as a vector or anything else like that. We assign row names given the tissue. So our rows will have a tissue name. Our column names aren't the columns of our matrix won't have any name. Uh, there's this whole palette of making colors in R, and it's a whole package itself. It's called the R Color Brewer. It's its own library. If you want to use it, I think you have to install packages for it. I don't think it's built in. I don't think it's native to the R base. I think you have to add it. But either way, this R Color Brewer lets you design a palette based off of a single color. And so what it's doing is just generating 9 colors that are blue, right? And it's going to be in this kind of heat map fashion. And then from there, we're just using this Pheatmap command. So a heat map, visually, we're going to look at a better heat map in a minute. But let's just kind of look what happens whenever I plot this graph out. Pheatmap, because I don't have library Pheatmap installed. Library Pheatmap. Okay. Okay, so this is our pairwise comparison. So what we should recognize is that the same sample. So each box represents. So let me back up. So the column names are the same as the sample names for our as the row name. So if this row, we'll just say this is MV liver, that's JB liver, that's GC liver, etc. Our column names are also MV liver, that's JB liver, GC liver, etc. And so each of these squares is a measurement of how similar those two samples are to each other. So MV to MV is identical, so it's the most similar. Of course, it's like a similarity of 0. And so down this entire diagonal are basically samples compared to themselves. So that's JB compared to JB. That's GC compared to GC, etc. Um, and so this box would be a comparison between, say, MV and JB, the JB liver samples. So what this is really showing is that all of the liver samples are more similar to each other than they are to anything else, and all of the kidney samples are more similar to everything to each other than anything else. When we try to say, if we're looking at this box, this is a comparison between a distance, a distance between a liver sample and a kidney sample. It's a very big number. So how this is drawn out is that the dark blue numbers are very small numbers. So the darkest blue is a 0. Um, and the lightest blue are big numbers. So this scale range from really, really similar to very, very different. Um, and so this is, again, this is just this is something that you see semi frequently in Heatmap plots. Heatmap plots is samples directly compared to each other, or expression values compared to exactly to each other. And so I think that's the last built-in function or plotting function that I wanted to demonstrate to you. Um, and again, like this is really the only function that's really doing anything. The rest of these functions are just to make the plot. It's this dist t assay VSD is what's actually measuring the distances among samples. Okay, so built-in plots are nice. They're user-friendly because you don't have to be an expert at R in plotting to be able to visualize your data. And that always makes a really good package whenever you not only supply the tools, but also supply the graphing tools that can make novice users be able to use and analyze your package, right? So a gene expression analysis, the hardest part honestly, in all of this stuff, is just reading the manual, right? Just understanding what this manual is actually saying takes more time than actually doing it. So this same manual, it's definitely not everything's intuitive, but everything that I've plotted out in this script is more or less the distilled need to know stuff. Everything that I've written down is basically the stuff that you need to know to be able to make this stuff work. So 67 lines of code, that's not too bad. R scripts can get pretty big, so R is kind of we call these, we measure how many words are needed to do anything as a metric of verbosity or verbose. So anything that's really verbose takes a lot of words for anything to do. So like C++ is super verbose, Java is really verbose, Python's not verbose at all, awk is even more less verbose, and R is just somewhere in the middle. It does take a lot of lines to do anything, um, but it's not the most of any programming language. Uh, so, but let's but since I am good at R and I am pretty good at ggplot, I can make some custom graphs based off of this data, and so we will. Um, so I was thinking last night is like this is kind of like the equivalent of like watching Bob Ross paint, but with computer stats and um, uh, programming languages, right? So you won't be able to produce a Bob Ross painting, but you can at least be guided through and kind of see what's being done. If anything else, just to know that all this stuff exists. Anyways, but let's start a new script. We'll just call this plotting. What is it? I'll just call this expression. Save as. I need to save this one real quick. Library. And so I did all this stuff at night. Um, so this shouldn't be too bad. We need our ggplot library. We also need our, we're going to need melt in a minute. And so that's out of the reshape library. I'm importing that as well. All right. So first thing I'm going to do is import the expression file. So this is like, let's see what this file is going to be. So in one of our outputs, um, this. So we normalized our expression values based off of everything, and now we save that as a comma delimited file, and now I'm just going to re-upload this file. So that's what this expression is. So it's I'm just going to call it norm expression, norm exp, and so this is a read.csv. I think I've mentioned read.csv before. Let me know if I haven't. Um, and I need to set my row names. So my row names do have like my G names are the first column, and so I'm just going to set my G names as actual row names because having row names kind of makes ggplotting a little bit easier. And also, I'm also going to read in my statistics file. So my statistics file is the results of this command. So I have all of my p-values associated with this if this gene is differentially expressed, and I have them sorted out smallest to largest. So this is my stats file, and so I called this stats over here, but I'm going to call it something else. I'm going to call it stats de because stats is already defined. I think stats is already a defined function, or at least stat is. Just to make my life a little bit easier, make sure I don't upset R. I'll just call it something that's not RA defined. Read.csv. And so this is DESeq results. Again, my row names are G names. Oops. Save that, source it, make sure everything's going to load up nicely. Okay, no major problems. So my stats, if we look at my stats DE stuff, we look at this crapness. My p adjusted. I'm going to sort. This is already sorted, but I'm going to scroll to the bottom. And I have a bunch of these NA values. It just means like these there was a zero somewhere, or these values are just too similar, or the statistics just does not allow for corrected p-value, or it's just kind of garbage. So all of these rows that have an NA, they will cause an issue with plotting because I'm about to plot my p-adjusted values. but these NAs will make it, it won't stop me from doing it, but it will cause some grumpiness. So I'm going to get rid of these NAs. And so R allows me to remove NAs pretty easily. Stats, and so what I'm going to do is call this command called na.omit. It means that if any row, if any column, if any cell has an NA in it, it's like get rid of that entire row. So I'm going to do that. So that's going to get rid of all of my NAs. And also I want to define a variable. So I do have, my P adjusted. I know if my P adjusted is below 0.05, I know it's statistically significant. If not, then I know it's not. But trying to make ggplot do that without changing the table is a little bit tricky. It's less intuitive than what it already is whenever I change the table. So um, I am going to change the table and add a column to my table that's based off of a condition. And that condition is whether the p-value is below or equal to 0.05. And so I'm going to make a new column, stats de sig. And how I'm going to do this is using an if-else command. So R has this built-in if-else. And so an if-else statement is like, if this condition is met, do this. If not, do that. And so that's kind of describing, but what I'm saying is if the p-value is less than 0.05, then make a new column that has sig in it. But if that value is not below 0.05, make sure that column or that row variable will have a not or something like that. So it's if else. My stats dollar key adjusted is less than or equal to 0.05. It's sig or not. And it's just, again, we just have these two conditions. Sig will be in place if the condition is met. Not will be in place if the condition is not met. So conditional statements, these if-else statements are fundamental to programming languages. Most of them don't look like this. This is just an R-specific if-else function. But let me source that again, and I should now have a column in my stats. Sorry, DE. You gotta tell me when I'm doing something stupid. Stats DE. Hopefully no complaints. No complaints. And where's that? Stats DE. Okay, so now I have the sig column. So now I have at least my rows are defined, whether the condition was met, whether this value is below 0.05 or not. And so the first graph I'm gonna replicate is something we've already seen before. It's just that plot in a graph that we did on Wednesday. So all this plot is just the x-axis is a log transformed mean expression value, and the y-axis is the log 2 as is the what is the y-axis? It's a log fold change. So stats de. I need to ggplot this. So I'm going to ggplot this. So my x is so I need to log 10 this. Otherwise it won't look right. It's the base mean column. Base mean. And my y is, so this is the log 2 fold change. And so this is a dot plot. So I'm just going to save that real quick. I'm going to start manipulating down here a little bit. Oh, something's gone wrong. Come on, homie. Just give this one a second to get going. Just a lot of data points in here. There we go. All right. So, right, we've seen all of this before. It's just I don't have colors associated with my p-value, right? Um, but I added that nice little column so I can go ahead and add that. So I can change the color in my, need another thingy there, need another rotation. So I'm going to add a, I can add my color variable. Um, so let me just do that real quick. Yeah. Let's just change two little things about this plot MA. I'll change this. Color is equal to sig, so that new column that I added, whether it's significant or not, GG point, and I'm also going to get rid of the legend that would appear whenever I do this. So this is theme, legend position is gone. Stop. Okay, almost. Almost. Come on, homie. You can do it. Why? Maybe fill? What'd I do? I did this before. What's wrong? Something stupid is happening and I don't see what. Just try it one more time, just for the hell of it. Actually, I know. I think I know. I think I know. I got my. It's always something so simple as a parenthesis is in the wrong place. That's all it takes. It's just a parenthesis in the wrong place, and it just won't work as well for it. Let's try that one more time. There we go. So my significant p-values are colored in blue, and my not so significant values are colored in red. So we've seen this before, right? So that's all that plot was doing. Is it was just reading those two columns and making a plot based off of the what a mistake. Um, so that's one of these. The plot MA. Let me make sure everything's added in here. Color is equal to sig and add my theme, change my theme up. What was my theme? Legend position equals none. Okay, so that's one plot that's not always that common, but it does help you look at your data. A plot that is common in biology, at least it used to be common, is something called a volcano plot. Um, this is not built in to GGplot or DESeq, but it is nonetheless, there are packages that'll make it, but this isn't that hard to make on our own. So I'm going to make this plot called Volcano. And you'll see why it's called a volcano plot in a second. But what this is it's the x-axis becomes the log fold change to fold change, and the y-axis is the p-value. But not only that, it's the adjusted p-value. It's a log transformed p-value. So our p-value ranges between 0 and 1. Um, and looking at that directly isn't very hard, but we can turn those into integers. Instead of these fraction values of 0.00 something, I can do a log transformation, specifically a negative log 10 transformation. And so that will turn these values into greater than 1 values. So they'll be turned into integers. Um, and let's see, log 10 p adjust. And so I want to color these based off of the significance. And this is a geom point plot. Geom. Let's see if this works politely. Why? Following aesthetics X and Y, I did something stupid. Because I did not say AES. Make sure my parentheses are in the right place. And here we go. PADJ. PADJ. All right, and so this is our volcano plot. Where am I? There we go. So my volcano plot. So again, the log fold change is on the x-axis. So this is between negative x and positive x. Anything to the right is going to be biased towards the liver. Anything to the left are these gene expression values that are biased towards the kidney. The p-values are, again, they're log transformed, and not only log transformed, they're negative 10 log transformed. And so it kind of looks like a splashy figure. That's why it's called a volcano plot, because it kind of looks like a volcano exploding. But again, not all volcanoes look like this. We have a lot of expression values that are different, and so in those cases, again, if we looked at the same tissue, you would most likely see dots kind of hovering in the middle with only a handful of them actually looking differentially expressed. But here it really just goes off in both directions. Again, anything to the left of the zero is going to be kidney biased. Anything to the right of the zero is going to be liver biased. And the highest, the further out on the y-axis, the larger the value on the y-axis is, the smaller the p-value. So we label this like this is point. A lot of this is going to be between 1 and 0.05 are going to be these red dots, and then the blue dots are p of 0.05 or smaller. Again, it's just log transforming it this way. Transforms the data and makes it a little bit easier to look at. Again, these are kind of common. Sometimes they'll even make another column, like a third color for any value that's, say, above a log fold change of some number, to be 2, 10, whatever. That is also common. Say that change anything to the extremes, the extremely differentially expressed stuff as like color it black or color it some other color. Anyways, but that's a volcano plot. That's another thing we can make by hand using GGplot. I have a couple more that we can do. So let's build an actual heat map of our data. So this one does take a little bit of magic. So what you're going to see is a little bit of our magic. I'll try to explain what I'm doing, but it is a little bit not intuitive. But first, so if let's say we just want to look at the gene expression values of the top 10 differentially expressed genes among all of our tissues, of all of our samples. So one, I need to get the top 10 uh, rows, or let's start with top 10. So I'm just going to create a new variable called top, that's stats de, and we're just getting the first 10 rows. Okay. And interestingly enough, since, let's see if we look at these values. So if we look at our matrices, if we look at our data frame, so that's not what I want. I want stats DE. So it has G names as the row names, as well as the uh, what is that plot? Which one is it called? Norm expressed also has the G names in the as the row names. So I can use this command called merge. It's not the one I want. I can use not the one I want. I can use this command called merge. That's going to merge columns, or sorry, it's going to merge rows of separate data frames into one data frame. And if that row doesn't exist, if that row name doesn't exist, throw it out. So I can merge my top and my norm expression that's keeping track, that has all of my expression values. I can merge them by by the row name. So that's by 0. That's equivalent to the row name. So I'm going to rename this as top. So top is just getting out the top 10 genes that are differentially expressed, and then merge is going to pull out the rows that contain the same gene name from my expression data. So let me save that. Hopefully that won't take too long to resource. Top. So let's look at top. Um, So my top has my gene name. Again, so it has also the same columns from my stats package or from my stats data frame. So I have the base number, the log2 full change, the p-value, adjusted p-value, whether it's significant or not. And then it also contains the actual expression values for those genes for every sample. Right, that's what merge did. I used that's what we did. But this gets a little tricky. Um, I don't want all of these columns. The only columns that I actually want in this data frame are the G names and the expression values. So this is definitely some magic that's about to happen. I'm going to call this Heatmap. I'll try to explain what's happening, but first of all, I'm going to define this vector called keep that's going to have all of the columns that I care about. It just has the column names that I actually care about. And I have this written down somewhere else, so I'm just going to paste this over so I don't have to write this whole thing out. But again, it's just the column names that I care about. It's the row names and the tissue or the sample names for all of my columns that I care about. It's just this variable that I'm calling keep. Now, the tricky part is kind of the weird thing that's about to happen is that I'm going to ask R to look through all of my column names and only keep the column names that are in this vector keep. Again, this is kind of a Googleable command. I don't know this one off of the top of my head, but it would be top. I'm going to make this into, make sure I'm calling this as a matrix. So again, my before the comma is my rows, after the comma is my columns. So I'm looking through my columns and I'm saying, are the names of these columns in keep? And so this is just a syntax for R. It's just a way of asking if something is in something else. Weird, but here we are. So I'm gonna call this top again. And if I do this command, that should get rid of all of these stuff that I don't want. So if I look at top again, something has gone wrong. Something has definitely gone wrong. Let me do all this stuff again. Maybe I can get all this stuff. Yeah, that's fine. Okay, so my row name, so all the other junk columns that I didn't want are gone, and now I just have the column names that I want, so the row names and the values for all of my tissue samples. And so basically what I'm going to do is assign a color to each of these values that's on a range from low to high. And I'm going to keep these rows and these columns in their place. Again, I'm just going to apply a color that represents this number. So let's see. But this is again some wonky magic, because I need this column to actually be a row name. In its current state, it's not actually a row name. It's just a value to the column row names. And there is definitely a difference between this and an actual row name. So I need to turn this into a row name. This is definitely not intuitive. This took some time to figure out how to actually do it and find the code to do it, but whatever. Um, Top, so I'm going to call top and -1. So it just means I'm calling I'm making a new matrix called top, or new top, that's everything except for the first column. This -1 basically means everything. I think this means everything but the first column. I'm pretty sure that's true. Let's double check. New top. Yep, everything but the first column. So I basically trimmed off the row names. But I want to make that I need to add row names. So I'm gonna add row names to my new top variable, that was from top, and it is just the first column. So again, this is just a wonky way of doing it. It's kind of roundabout. There's probably a better way, um, but nonetheless, you do what you have to do, and you do what works. So if I look at new top again, now I have my G names are the actual row names. Again, this is just stupid manipulations that we have to do to make GGplot happy. Okay, and so now let's actually, or it's not even GGplot, I'm using Pheatmap. And so this is going to be, I'm going to log2 transform this data. Actually, I'm not going to do it first. I'm just going to do the raw data first. And then a couple other things with some other variables. So those row names are really long, and so I don't really want them. So I can change this to show row names equal to false, and we'll see how this looks right off the bat. It's going to look terrible. I'll tell you that right now. It's going to look terrible. But this lets me talk about some other things. That doesn't actually look that bad. And it's not the worst thing. But either way, so again, these numbers are assigned a column, and each value is assigned a color that gets measured between 0 and the top number, and it gets scaled on this top number. So this box is still an expression value for a kidney. It's the first gene whenever AJ kidney, and it's low. And over here, it's higher. This red, this is the highest expression value among these 10 genes. And so this was in JB liver. And so what you tend to find, and what we're going to see, is that how this is going to work, since there's so many differentially expressed genes, we're only looking at the differentially expressed genes. Some are going to be higher in kidney, and some are going to be higher in liver. And so this matrix, it will be sorted out, and it's sorted out into this dendrogram. So these rows and these columns do get sorted to reflect these tissue differences. And so it's basically it's clustering the columns based off of what's similar to each other, and it's clustering these rows based off of what's most similar. And so these 3 rows are more similar to each other than the others, and these 7 rows are more similar to each other than the others based off of these expression values. So one thing that I kind of want to explain to some degree is transforming data, doing this log transformation data. So let's look at this in Excel because Excel is a little pretty quick about it. So I'm just going to have all these expression values that range from really small to really large, and I may even go take this out further. Let's add a few more data points. And so if we try to draw a heat map or we try to compare these values to each other, it definitely gets tricky because of the variance is so wide in these samples, and these values are effectively already on a log scale. So let's see what happens when we actually log2 transform this. So I'm just going to log2 transform all of these values, and we'll plot these out. We'll plot these out in a nice dot plot. Okay, and so more or less what it's doing is that it's smoothing these expression values out. It gets this, it makes our values much more comparable and easier to look at, and also puts everything more or less within the same order of magnitude on the same scale, so that a change from 3 to 5 would be the same change as going from 30 to 50, and it's measured as the same change of going from 300 to 500, 3,000 to 5,000, etc. All of this becomes these orders of magnitude become much more similar to each other. So whenever we have such a wide range of expression values, we almost have to log transform them to be able to look at them or do any kind of to play around with them. So let's see. Let's do this name again. Let's do this heat map again, but log2 transform it. But there are zeros in here, so I need to add a 1 to make sure that everything looks nice. So whenever I log transform it, now everything gets much easier to look at. It gets a little bit more comparable. The scale, our color scale gets smaller. Our numbers, the range in our numbers gets smaller. So now it's much easier to see where the genes, which tissue the genes are more expressed in by doing this log transformation. And a heat map is really common in expression analyses. It's one of the easiest ways to display differential expression. And even whenever we change this, whenever we changed our graph, whenever we log transformed it, again, it makes data a little bit more comparable to each other. We had 3 rows that were similar to each other, but now at this point, now we have a 4th row is added after we log transformed, after we made this a little bit more comparable to each other. So let's expand this out one more time. Let's change. I'll leave this here. I'm going to save this code and source my file. I don't think it'll take too long to redo all this stuff. Okay, but let's look at, say, the first 100 genes. Instead of looking at the first 10 genes, let's look at the first 100 differentially expressed genes. Let's see what that Pheatmap looks like. Oh, it's terrible because I didn't log transform. Stupid. Log2 plus 1. Yeah, this is why we need a log transform. When we log transform it, hey, look at that. It looks way nicer. Looks way nicer after we log transform. Much easier to look at. Yeah. So you can definitely see, again, everything is going to get, we're going to cluster everything. All these rows are going to get clustered together. These columns will get clustered together, and we can manipulate Pheatmap to do that. We can make Pheatmap make this look however we want it to look, right? We can change our color scales. We can get rid of these dendrograms if we wanted to. But let's try it one more time. Let's look at what the first 1,000 genes looks like. It will still effectively be the same thing. We're just adding more genes to it. So let's just try this one more time with 1,000 genes. In adult, so right, yay. Same thing. Some are expressed in liver, some are expressed in kidney. No surprises there. But all of the liver samples cluster together, all of the kidney samples cluster together. The genes that are more expressed in the kidney or in the liver, they cluster together. All the genes that are more expressed in the liver cluster or kidney cluster together. Right? It's just it's a heat map. So heat maps are pretty common. Let's see. I think I only have one more. I have one more file to look at. One more way to look at this data. Let's just do one more plot, and we'll call it good. So we're gonna look at the individual expression values of the top 10 genes. So instead of looking at as a heat map, let's directly compare these values for each sample. Since my new top is already made, um, and it's in the way that I want it to be, so that GGplot is happy with it, I'm just going to get the first 10 rows out of that, and so that should be my first 10 differentially expressed genes. So why is that complaining? Because I think it's just not doing that. Okay. So I'm going to source this real quick. I'm going to block that out so I don't run it all the time. So top 10, right? We've seen this before. It's just expression values of each tissue for the top 10 most differentially expressed genes. Okay. Let's see. So, but this data frame as is isn't very polite for GGplot. It is nice for an XY plot or something like that, but I want to make not quite a histogram or not quite a box plot, but I need my data to be in the same format as if I were making a box plot. So I have to melt my columns together. So I'm going to make a new variable called top 10 melt or top 10 m for melt. And so I'm going to melt this all. I'm going to melt all of these columns together into one row that gets defined. Melt top 10. So I need to make sure this is as a matrix. I forget why top 10. I think it's just for my row names. I'm going to run this in the terminal here. Top 10 in. Yep. So now I have my column. Right. I melted all of my columns together, so I have my column name and an expression value. So for this gene, that's my expression value. Right. We've done this before. We've used the melt command before. Hopefully this shouldn't be a foreign concept. Okay, now what is about to be a couple of foreign concepts is adding another column. But first, I just want to change my column names because x1, x2, x value aren't very descriptive. It's kind of it doesn't help me very much. So I'm going to rename those to make my life a little bit easier downstream. So I'm going to change those names to gene. Library, lib, library could be sample, and exp for expression. Run that command. So names of top 10, right? So that got changed to gene lib expression, so that makes life a little bit easier. And now for another magic. So I want to add another column because even though I have these kind of have tissue in them, I need a specific column that tells me whether this library came from a kidney or came from a liver. So I need to make that column, and that column is also not intuitive. I had to spend a lot of time getting a function that would do it. And so the function that I found is this if-else command. We've seen it before. And this derivative of grep-L, or it's a derivative of grep, really. So R does have some overlap with Bash, and so again, I said this whenever we introduced it. We're not done with grep, and we're never going to be done with grep. Grep is like being able to read in computing. Grep never goes away. But it is renamed, so it's grepl. And so basically, what I'm doing is I'm going through each row of my lib column and asking if liver is there. If liver is there, create a value in my tissue column that's liver. If that liver word is not present in that cell, make it kidney. Again, not intuitive. Sometimes you have to Google around for hours to find a command that does what you want it to do, but let's see, that should change it. So now we have a tissue that has liver or kidney. So understanding what I'm doing isn't that difficult, but the code itself is always a little intuitive. We're almost done. We're almost done with this. Okay, so now my data frame is in the way that I want it to be. I have my tissue labeled, my library labeled, I have my gene names labeled, and everything else. So now I can start writing my GGplot on this stuff. So I'm going to make my variable, my plotting variable called expression or gene express, GGplot, top 10M is my data frame. 8x, x is equal to the tissue. Y is equal to, I'm going to log transform this again, log 2 of the expression plus 1 because there are zeros in here. And color by tissue. Seconds, color is tissue. Okay. Okay. So this is a genome point graph. We'll see. And so what I'm going to make individual plots. So I'm going to use my facet grid to give me individual plots of these points for each genes. That's gene. So let's just look at that real quick. Okay, so again, each bar is the gene, and these plots are colored, or these dots are colored based off of what kind of library it came from. Red dots are from the liver, blue dots are from the red dots are from the kidney, blue dots are from the liver, and again, you see in these first two graphs, the liver, this gene is more expressed in the liver than in the kidney, but in the remainders, expression is higher for the kidney in the remaining 10. So that's just a nice way to look at individual genes. You can pull out a single individual gene if you wanted to just look at the expression, compare the expression of all your samples for one gene. But GGplot lets us look at multiple genes all at once. Unfortunately, this gene name is so long that it really doesn't fill out this this panel very well. So whatever, but you still get the idea of what we're trying to do. I'm going to add a couple more things to this just to make it look a little bit nicer. I'm going to get rid of that. I'm going to get rid of the legend. None. And I'm going to get rid of this x-axis title. So I don't need my tissue explained. I don't need that there. So I'm going to get rid of that title. Element blank. Done something stupid. Mistakes were made. Mistakes were made. Okay. So if anything else, it just kind of winds this out a little bit. So and so I have my I don't have that that my X is tidal. I on my legends, I don't need those presently. I mean, I can interpret what's happening without those two variables present. Um, so yeah, so you can do a lot of stuff with right. I don't know, but I think you can definitely cheaply do a lot of molecular biology on the computer. You can do I think you can do a lot more molecular biology very cheaply than you can in a molecular biology lab, right? So hopefully you've learned something very different, or at least reinforced what you already knew um, about transcription, translation, and measuring differences among DNA, whether that's sequence differences or expression differences among tissues. Um, and I don't know, maybe I you got to remember, so like this kind of analysis really wasn't even available until like 2007, right? Prior to this, nobody is doing any of this. It's all by hand, very tedious lab work to measure, doing a lot of qPCRs to measure individual gene expression. But now we can do it very quickly. Um, and some people would like maybe not even these top 10 genes, but you may have a specific set of genes you're looking at. So some people may be involved in some ATP synthesis pathway and look at differential expression in that of those genes, say in different aerobic environments in hypoxic conditions or something along those lines. So we don't have, I mean, this was just an example. Picking out the top 10 is the easiest, but others may be looking at specific genes within a pathway, pulling those out and comparing expression among tissues or among experimental treatments and things like that. So you don't have to listen to me talk anymore for the rest of, well, possibly your lives. And I hope this was somewhat enjoyable for y'all, and I'll work on a final and get that released over the next either today or over the weekend. All right, have a good one.